Pyruvate oxidation takes place in the cell membrane. Each of the two pyruvate molecules from glycolysis will be oxidized into a molecule of acetyl-CoA. In this animation, for simplicity, we are only showing one of the pyruvate molecules going through this reaction. First, in the presence of oxygen, the pyruvate loses a carboxyl group to create a CO2 molecule. This CO2 will diffuse out of the mitochondria, out of the cell, and into the environment. In other words, it's the CO2 we exhale. Then, NAD+ bonds with an H+ molecule and is reduced to form NADH. These NADH molecules will then go to the electron transport chain. Finally, the remaining carbon from the pyruvate is joined by a CoA molecule to form acetyl-CoA. Since there are two molecules of pyruvate, two molecules of acetyl-CoA will be produced. These acetyl-CoA molecules will move on to begin the citric acid cycle, also called the Krebs cycle. The citric acid cycle begins with one of the acetyl-CoA molecules from pyruvate oxidation. Acetyl-CoA combines with a 4-carbon molecule, oxaloacetate, to form a 6-carbon molecule. This is the step that will always begin the cycle. The CoA molecule is ejected. Next, one carbon is removed, creating CO2, and a molecule of NAD+ is reduced to NADH. Then another carbon is removed, creating CO2, and another molecule of NAD+ is reduced to NADH. ADP is introduced, and through rearrangement of bonds, one ATP is produced. We now have one 4-carbon molecule. Next, this 4-carbon molecule is oxidized twice. The first time it reduces FAD to FADH2. The second time it reduces NAD+ to NADH. This process regenerates oxaloacetate, which is used to start the cycle over again. Now, the process begins with the second acetyl-CoA. Each cycle produces 3 NADH, 1 FADH2, 2 CO2, and 1 ATP. Since there are 2 acetyl-CoA molecules, the CAC results in a total of 6 NADH, 2 FADH2, 4 CO2, and 2 ATP. With pyruvate molecules in hand, UniVirtual students journey to the mitochondria to continue cellular respiration. Navigating through the intricate environment, they collect essential molecules such as CoA for oxidation and NAD+ to store high-energy electrons. Once again, they access their PAX screen to combine these components, successfully transforming pyruvate into acetyl-CoA. Excess CO2 is discarded, while NADH is safely stored for the next stage of respiration. Inventory management is critical, as the student ensures they have enough space for the next phase, the citric acid cycle. With their energy stores growing, they press forward into the mitochondria's depths, ready for the next biochemical challenge.